Welcome to my little video presentation with the title What Will Happen if Garbage Collection is Not Proper Working in Our Brain. Here you see in principle two neighboring cells, and in these cells daily new protein will be synthesized, and I will show now for the example of alpha-synuclein what will happen when the protein is overloaded by protein production. Alpha-synuclein is a small protein, but nevertheless it's important because it's thought to be involved in the development of Parkinson's disease. Once alpha-synuclein has been produced, it will be detected as monomeric protein, but as well as dimeric protein because it has a high tendency to form these dimers. So over the time, the cell will produce new alpha-synuclein protein because protein get fast, let's say, aged. And so under a certain situation, the cell has to get rid of this old age and overproduced alpha-synuclein protein. In principle, there are different systems. The first system I will mention here is the so-called UPS system. By tagging aged proteins by a ubiquitin tag, the protein will be degraded. With simply words here, this means you just wrap the old alpha-synuclein protein with toilet paper and you trash it into the toilet. So the problem is solved. But what will happen when the toilet is blocked or occupied? Then, of course, there's a second system, which is called the lysosomal system. So in simply way, protein will be trashed into this garbage bin. This garbage bin can be set on fire, and so the material can be burned down. So Parkinson's disease patients showed very often deficits in both of these systems. So the big question is coming up. What will happen when both systems are not working? Of course, the cell will be overloaded with new synthesized and old aged protein, illustrated here by these full garbage bins. Alpha-synuclein will also be stored, let's say, in this situation in a different conformation, and so the generation of so-called alpha-synuclein fibrils can be detected. Finally, when this process is not stopped, we can also see the formation of so-called Lewy bodies, which are very predominant in the cells, as illustrated here by the generation or formation of these big hills full of garbage. This is a very critical situation, and of course, it can turn the cell into cell death. So there's not a big wonder that the cell tries to escape and is performing an alternative way. By generation, so-called exosomes. Exosomes are little protein vesicles or protein bubbles, which means that vesicles are filled up with old protein material. These bubbles can be released very easily from one cell, but they can also be easily uptaken from the neighborhood cell. And here the problematic question is raising up: what will happen now when the neighboring heart is uptaking this kind of old material? So will it also induce there a kind of pathological process, as we know from Parkinson's disease patients? This is the main question and our main focus yet. Thank you a lot, and bye-bye.